Hello, my name is Erik Hjerde, and I will be giving this theoretical introduction to the hands-on exercise that you will be given later. So it's an introduction to quality control, filtering, and the assembly of metagenomic data, and finally validation of the assembly. Just a quick recap on DNA sequencing. So normally we will sequence a DNA fragment from both ends, producing read 1 and read 2 here, where you have a certain distance between them if the fragment you're sequencing is long enough. So you will have a forward read and a reverse read covering most of your DNA fragment. And well, this is also a recap from previously. So the base calls are made on the Illumina machine directly from the intensity of these colored bases that are incorporated while the DNA strand is being produced. And we can measure the intensity and then also the quality, or call the bases and give them a certain quality based on the intensity. So the sequencing technologies aren't perfect, and we will have some errors in those sequences, and we are doing a quality control of our raw sequence data because we want to discover these errors and at least know they are there, or deal with the data accordingly. And I'll get back to what we can do with data that are of lower quality. So when we do quality control, we can look for low quality sequences, of course, sequence contamination, and also if there are any bias in the raw data, which can be caused by some technical problems with the sequencing machine and so on. This is how a FASTQ file will look like. So this will come directly from the sequencing machine, and it contains 4 lines per sequence that has been produced. So the first line of each sequence read starts with the @ sign and the name of the sequence. And then the second line will be the DNA sequence of this sequence read. Third line will be a line separator, and the fourth line will be the sequence quality. So you can see the length of the fourth and the second line is equally long. So each of these letters and symbols represent quality of the base that has been called. Okay, and normally these FASTQ files can contain millions of sequence reads, so we are only viewing a really tiny fraction of the complete FASTQ file. So the quality scores may differ depending on the technology being used to produce this. So nowadays most, at least the Illumina machines, will produce quality scores using a scale or called 1.8 or Fred 33. But if you're working with the older Illumina data, you should check if there is another quality score system being used for that. So just to explain this quickly, so you can see in 1.8, Illumina 1.8, so the scale starts at the ASCII letter here, and it contains these letters up to, so there's 41, maximum score is 41, so that's the letter I. So everything above 30 is regarded as really good quality, and that starts in the letter at. This is a good score. So all letters in Illumina 1.8 are really high scoring base calls. But if you are using Illumina 1.3, for example, you see the letters here. This scale starts in the at position, and so all the letters will be really poor quality reads, and the capital letters and the small letters over here will be good quality scores. So it's good to know what kind of system that has been used on your data. Yeah, so Q30 is regarded as everything above Q30 is regarded as good quality, and that means that the probability of finding one incorrect base call per the probability is to find one incorrect base call per 1,000 nucleotides. So this is everything above 30 is regarded as really good quality. When we perform quality control, we can generate a lot of information from the raw data. So normally you will get a general summary of your data, summarize how many sequences you're dealing with, the average GC content, the sequence length distribution, so from 35 to 301 here, and so on. So lots of useful statistics here. And also, I mean, the length distribution of your reads in total can be, this information can be found, and it should be, of course, close to what you expect from the sequencing library you produced. You can also view the average quality score in each position of your sequences, and normally you will see that the quality scores drops towards the end of your sequences. That this is a quite normal phenomenon, and so now we're looking at the forward read of a paired end sequence library that's been sequenced. The reverse will actually have an even more dramatical drop in the quality of read towards the end of them. Yeah, we can also view things like the average quality score across the whole data set. So you would like most of your sequences, so on the y-axis here you have the number of sequences, so you would like most of your sequences to be in the top end here, above 30, the average quality score of each of your read in this region here, and not down here. So yeah, these quality controls can also look for sequence artifacts. So in the sequence reads, you would expect that you have an even distribution of the nucleotides in each position of your reads, but in some cases you will see that you have a bias in some positions, especially in the beginning of reads, and this could be due to that there are sequence adapters that haven't been removed, for example, that will introduce a bias in some positions. Also, very often when you see this in the beginning of the reads, it could be due to, so Illumina has written informed the users that this is due to that the sequencing machine itself needs the first nucleotides to calibrate its reading the intensities of the base call. So it's using the first sort of cycles in the sequencing reaction to calibrate the machine itself. But anyway, these are artifacts that can be removed, as we shall see later, and you will also do this in the exercise. Yeah, duplication levels is also possible to detect, and ideally you wouldn't like the reads to be duplicated. So you would like to have as many unique reads as possible. If you have lots of duplication, that could indicate that you have that there are some biases in your amplification, or the level of unknown bases can also be visualized, and so on. So I've just listed a couple of popular quality control tools that are used in this course. You will work with a tool called FastQC, which has a web graphical user interface, and it can take both Illumina and PacBio sequencing technologies as an input. Okay, so why do we want to do quality control and filtering and trimming of the data? Well, I mean, the most obvious thing is that you want to know your data set, and you want to remove poor quality data, as it may introduce errors later in your analysis. So there's you can either filter the data based on a certain threshold. For example, you want to have only sequences that are longer than a certain length, or that has a higher quality score than a certain threshold, or you can actually trim each sequence read based on some criteria. For example, if you see that most of your reads have they drop in quality towards the end, then you can either, for example, remove the last 20 base pairs of each sequence read, or you can set actually a threshold saying that a sliding window of 5 nucleotides when the sequence quality drop below Q30, then trim the sequence there. So there's lots of options using different tools that are available. Yeah, so in some cases, the first slide I showed was a DNA fragment where we sequenced the fragment from both directions, where you had an inner distance, a gap between the forward and the reverse read. In this case, the insert, the DNA fragment, is so short that the forward and the reverse read will overlap, which is okay in some cases. There's nothing wrong with this. That will happen. In some cases, you will have reads that are DNA fragments that are really short, so both reads will actually read sequence into the adapter in the 3' end, and in those cases it is that will create these biased positions, which we saw in an earlier slide, and you can also see it here on the right hand side. Because so the sequencing machine itself will remove the 5' adapter. It has built-in tools to remove the adapter in this end, but not when you enter the 3' and adapter. So that is something that you would have to remove manually if your library really contains a lot of short fragments. Yeah, you also, if you have sequence reads with quite low quality at especially towards the end like this, you can trim the reads that from a certain position, as I tried to explain earlier. Or you can also trim all reads that has, or filter all reads that has a lower average quality than a certain threshold which you set. Here's a list of some tools that are available for trimming. In this workshop you will use the Trim-O-Matic tool, but there are also a lot of other tools available which are quite popular. So I'm just gonna mention very briefly that there are other ways to do quality control also. It's possible to do quality control using Kmer analysis. We're not going to do that on the course, but just to mention it. So a Kmer is it's a subsequence of your longer sequence. So in this case, this 7-mer is the Kmer is 7-mer, and you share the original sequence into 7-mers, or using a sliding window of 7. So you produce a lot of 7-mers from this sequence in red here. These are Kmers. And it's possible to use Kmer to give you an idea of the quality of your sample. It is, for example, for genomes, it's possible to use it to detect contaminations if you have two species in there or more. This is because each genome has a certain portion of unique Kmers in them, so they will appear very few times. And on this plot here is the Kmer spectrum, so each Kmers are the occurrences of each Kmer are plotted. So you have the count here and how many Kmers on the y-axis, and the number of occurrences of these different Kmers. And you can see that some of them occur very frequently and some of them occur less frequently. So this is what you would expect from a single genome. From metagenome, you're sequencing maybe 1,000 of different species, and so you will not expect this distribution of the Kmers. You will expect a more even distribution of your of the Kmers. So if a good metagenome will have a more even distribution. Trying to illustrate this here. So Kmers can be used also to say something about the quality of your DNA. Just to mention that each species will have, if you do a Kmer spectrum analysis, each on specific genomes, you will see that each genome have a particular Kmer distribution pattern. This is shown here, that each of these species that are listed on the left-hand side here have a unique distribution of Kmers relative to the others. It's possible to do this Kmer analysis on complete genomes and even on metagenomes you can actually do to look at the distribution of species in your sample from this. Okay, and next question is, do you have sufficient data? So working with metagenomics, it's always a question how deep you should sequence in order to get the information that you actually want to have. The newest Illumina machine, the NovaSeq, produces up to 20 billion sequence reads in one machine. But even though this is an enormous number of sequences, it is still far from capturing all the DNA that is found from every microorganism in a particular environment. And in any kind of environment, the abundances of the different microorganisms will vary a lot. So you will have some species that are present in large amount and others that are barely visible. Yeah, have been done studies to see how much you actually need to sequence in order to cover complete genomes of certain species. So, but in this figure I've put at the bottom here, I'm trying to illustrate that some species, for example, in this case C and F, are present in large amount in this sample of 7 different species, while others B and D are present in very low amount. Okay, it is possible to sort of estimate how much sequences you need to produce in order to cover to meet the goals you have set for your sequencing. So, for example, if you want to sequence a certain number of species with a relative high abundance, you can calculate that. But and there are some tools. There's a link to one one paper describing this technique. But the drawbacks of this is that you actually need to know the taxonomic profile. So you need to know the relative abundances of each of the species in the sample. So how many percent of E. coli in the sample is it, and how many percent of another species is it? So this is information you need to have in before you can do this calculation of or this estimation of how deep you need to sequence to meet your requirements. Okay, I'm gonna move on to sequence assembly. We have been through sequencing in another talk and quality control in this talk. Now we are here at the assembly. And so the assembly is the process of putting all the sequences together into larger fragments. Normally, if you've done a quality control and filtering, you will probably like to do it on the sort of the good quality reads that you obtain from this. De novo assembly is the most common method to do it. So that is when you sequence or try to put together these sequence reads without really knowing how it should look like in the end. There's also a possibility to use a reference and try to use the reference as a map to put certain parts of a genome, if it's a genome you're working with, in sort of the correct order. But for metagenomes, you rarely have a complete reference genome, so it's de novo assembly that is the most common thing to do. Now, doing de novo assembly is really challenging if you don't know what the result should be. So for a genome, so this is an example I'm trying to illustrate with a genome. So what you do with the genome is that you're taking multiple copies of so you're taking many cells, isolating the DNA, sharing it into pieces and assemble it into one chromosome, or so that is what you would like to do with the genome. But if you don't know what the carriage should look like, you might end up producing this from the assembly and not actually knowing that you have done a slight mistake. For metagenomes, it's even more challenging because you're not only dealing with one genome, you're dealing with it could be thousands of genome that you are cutting into small fragment, and you are trying to get back to or assemble them into in the correct order again, and for each individual. So it's quite challenging. And again, if you don't know what it should look like, then you might end up putting together pieces that shouldn't belong together, and so on. And why do we want to assemble the genomes? Why don't we just work with reads or doing amplicon sequencing if we are interested in knowing what sample? Well, I mean, if we sequence random metagenomic samples, so we're sequencing, we are trying to sequence randomly from a sample, so any kind of DNA that's in the sample will theoretically be covered by the sequencing, especially if we sequence deep enough. So theoretically, we will be able then to put together all the genomes that are in the sample if we sequence deep enough, and that of course will be really important in order to discover new species, that especially things that have been impossible to grow in the lab and has never been sequenced because they are impossible to grow in the lab. These are things that we can obtain by doing random sequencing of metagenomes. I think that is one of the most important arguments for doing complete genome sequence. And of course, you will get other types of information than you would do with only 16S amplicon sequencing. I mean, you can dig more into the functional parts of this community that you are sequencing, but that will be covered in a later talk. Yes, there are a lot of tools available for doing assembly of metagenomes. In this exercise, you will use a tool called MegaHit, which is a really fast and very accurate, surprisingly accurate tool. Metaspades is also a nice tool to do. And a couple of others here are quite compute demanding. So we will stick to MegaHit, which is a really fast tool to run. Once we have assembled a metagenome, there are actually very few ways to check if you have assembled it correctly. This is not an easy task. So there's a tool called MetaQuast that can compare the assemblies between from different alignments. So normally I would recommend that when you do assembly of a metagenome, you should probably pick more than one tool and assemble it with different tools, and then try to compare the assemblies against each other. Now, MetaQuast is really nice in the way that it can also if you feed it an assembly, it will try to look up what kind of species that are in your contigs. So it does a quick blast of your contigs and try to identify from which species the contigs are from, and then it will build up sort of a library of all these species that are present in your assembly, and download it directly from NCBI and sort of create a huge reference metagenome from this, and then it will start to map all the contigs onto this sort of pseudo metagenome it created, and from this it can create some statistics that will help you in evaluating which method or tool is best to use. So on this slide here, it's from the MetaQuest homepage, and what this does, what you see here are 4 columns which are from 4 different assemblies. So you recognize Spades here, Ray here is another metagenomic assembler. So so here we have 4 assemblies, and MetaQuest has compared them against this pseudo reference genome that it obtained by taking all the contigs. So if we look at the Spades assembly here, there are let me have a look here. Number of contigs 40,546. So it looked up this which species these contigs are likely to come from. So from probably a lot of different species. So, and it has downloaded all of these species from NCBI. By default, I think it only downloads the 30 most the 30 sort of genomes that are present in most of the contigs, which is, yeah, I think that's the default. Anyway, so from 30 genomes, it has, from these 30 genomes, which it creates this pseudo meta genomic reference, it will map all these 40,546 contigs on top of it, and then it will produce all these statistics and saying, trying to see how many of them that doesn't really look like the reference. It doesn't mean that it's wrong, but it's not identical to the reference it's actually downloaded. So lots of statistics here that will help the user in the end to evaluate if one method could be preferred over another here. So you will have the chance to run MetaQuest on your assemblies in the exercise. Yeah, and then of course, you will also try to run the assembly on quality trimmed and filtered data and on raw data, and you can compare which produced the best assembly from that. Yeah, and yes, and put this slide in here just to show you that we have used MetaQuest in a study to show that when you do random metagenomic sampling, there will be a lot of 16S RNA sequences in the among the sequence reads, and these are very conserved genes among species. So it's, for an assembler, it could be very hard to separate two 16S sequence reads from each other if they are from quite closely related species. So they will sort of can create errors in the assembly, and they also makes the assembly more complicated. So by removing all the 16S RNA sequences from the sample before assembly has a very positive effect on the assembly. It will in all cases produce better statistics if you remove the 16S RNA reads from the sample. So that's just nice to know if you're working with random sequencing metagenomes that's been sequenced by random sequencing. Yeah. So the exercise, which will be available to you, can be summarized like this. You're going to work with samples that's been taken from baby feces, and the first part of the exercise would be to use to get to know the sort of the Linux environment and how you maneuver around using the command line, you will be quite familiar with FASTQ file format. And after that, you will start to do quality control using this FastQC program. Then you will merge reads that are overlapping, you will trim reads that has poor quality, and assemble at least one metagenome. And in the end, you will compare this with some pre-made assemblies that we have done on the same data set that you will be working with. So, good luck to you.